So we'll continue with our discussion of skeletal muscle by looking at a muscle fiber, a single muscle cell and the different parts. So the word sarco means flesh, and when we're referring to skeletal muscle, the prefix sarco is used a lot. So the cytoplasm is called the sarcoplasm, and it's just typical cytoplasm. It has organelles, it has all the components that cytoplasm normally has, with some specialized organelles. There's also they are multinucleated in that they undergo mitosis, but they don't undergo cytokinesis. There are many copies of the nuclei, and that's because the cells are so big. You can have a single cell that is 50 centimeters long. Like they can be huge. And so you have to have protein synthesis happening along the entire length. It would take too long to distribute a protein made centrally to the periphery. So there are multinuclei. There's a sarcolemma. And so this is the plasma membrane. This is a typical cell membrane with phospholipids and cholesterol and embedded proteins. But there's a modification that we don't see in any other cells. Because these cells are very large, there's invaginations that pass along these tubules. They're called T-tubules, transverse tubules. And so they bring the surface and the cell membrane into the center of the cells. And this is for coordination, and so signals reach all parts of the cell more or less simultaneously. We'll see in the picture here. So when we look at the picture, what we're seeing here, it looks like this stack of round organelles, which it is, and those organelles are the organelles of contraction, and they're called myofibrils. And we can see where right here, where the sarcolemma is rolled back, that there are these holes, and the holes line up with these yellow things here called transverse tubules. And you will see that the transverse tubules extend from the outside and they go all the way down, and they surround each one of these myofibrils right here, like that. And they do it in an organized pattern. On either side of the T-tubule, there is this modified sarcoplasmic reticulum or endoplasmic reticulum that we call the sarcoplasmic reticulum. And they have what's called the terminal cisternae that are located on either side of the T-tubule, the transverse tubule. The sarcoplasmic reticulum is basically a bag for calcium. The lumen of the T-tubule is outside of the cell. So that means interstitial fluid and the chemical properties of the interstitial fluid extend right down into the center of the cell in the lumen of each T-tubule. The T-tubule is made of the same phospholipid bilayer with proteins that the rest of the cell membrane is. We're going to see in a minute how this works. There is a membrane here that's made of the same phospholipid bilayer that is a bag for calcium. And there are calcium channels located all over it. We call this the triad because there is a terminal cisternae on either side of a T tubule. So the organelle of contraction, the organelle that actually generates the force of contraction, is called a myofibril. And basically the fiber, the cell, is filled with these things. They are bundles of filaments, myofilaments, because they're muscle, and they're enclosed within a sarcoplasmic reticulum as well. Because it wraps around the myofilament. So the sarcoplasmic reticulum, like I say, has the terminal cisternae. It looks like smooth endoplasmic reticulum. They have calcium pumps that bring calcium into them and sequester the calcium. They keep it away from the rest of the sarcoplasm, the cytoplasm. And they have calcium release channels that release calcium when they are signaled to do so. So basically, they are places to sequester calcium when we need the calcium out of the cell, cytoplasm, and release it when we want calcium in the cytoplasm. So these things are made up of, these myofibrils are made up of a repeating structure called a sarcomere. And the sarcomeres are made up of different types of filaments. There's two fundamental types. There are thick filaments that have the protein myosin in it. And myosin is part fibrous and part globular protein, so it ends up looking kind of like a golf club with two heads. And there are thin filaments that look like twisted Mardi Gras beads, strings of pearls. Each bead on the string is a protein called actin. There are two other proteins involved with the thin filaments, tropomyosin and troponin, and they are regulatory proteins. So if we look at the thin and thick filaments, one myosin molecule is this twisted tail and two heads. Now, on the heads, there are two binding sites. One binding site will bind with actin. The other binding site binds with ATP. These things are twisted together in a rope with all the heads sticking out. So it looks kind of almost like Velcro. What's interesting, from the midline of each filament, these ones extend to the left, these ones extend to the right, and the heads stick out. The thin filaments are this double twisted string of actin beads, and then this kind of string that runs over the actin beads, winding around, is called tropomyosin. And it acts basically like a rope. And now you'll see that every one of these little beads has got a spot on it that's called the myosin binding site. If the myosin binding site is exposed, it will bind to the actin binding site on the myosin. The two will bind. What prevents that from happening is that tropomyosin is in the way. This protein here, which has a quaternary structure, is actually made of three globular chains, and it's called troponin. Troponin has one part that binds to the actin, one part that binds to the tropomyosin, and a third part that has a binding site for calcium. And now these things are organized, these myofilaments are arranged in something called the sarcomere. At both ends of the sarcomere, there is a disc that kind of looks like, those egg cartons that they use in restaurants, where the in on one side is the out on the other, and so they can interdigitate. Z discs look like that. Picture a circle, a disc cut out of that, and so with projections sticking out on one side, mirroring an indentation on the other, and the indentation on the one side is the projection on the other. And so when we look at them or sideways, they kind of have a zigzag. So that's why we call them Z discs. And this becomes the anchors for the thin filaments. We're going to look at how this is arranged with the thin and thick filaments, and gives kind of a banding look. The striations that we see, the stripes that we see in skeletal muscle. There are I bands and there are A bands. And we should just go and look at the picture. I'll come back to this. This is probably the best picture to explain this. Okay, these are thick filaments. These are thin filaments. The thin filaments, you'll note, are attached to the Z disc. And they extend into this sarcomere over here and this sarcomere here. So the Z disc is kind of a part of both sarcomeres. So when we look at the thick filaments, they are obviously going to be darker. There's a lot of space. This little coiled is called connectin. It's almost like a little spring that allows the sarcomere to return to resting length when contraction stops. But if we look at from here to here, this will be quite dark because of the thick filaments. And so we call this the A band. From here to here, it goes thin filament, thick filament, thin filament, thick filament, thin filament, thick filament, thin filament. So it's very dark here and here. In between the thin filaments, it goes nothing, thick filament, nothing, thick filament, nothing, thick filament. So it's a little bit lighter. So we call the zone of the A band that's a little bit lighter the H zone, which stands for helios, which is the same word as halo It means a brightness. And then right in the middle of the H zone is something called the M line, the midline. And it's really kind of where the thick filament myosin proteins change direction. So we have the entire A band with the H zone in the middle and the M line down the middle of the H zone. Now, over here, with the Z disc in the middle, we have an area that goes thin filament, space, thin filament, space, thin filament, space. So this is a light colored space. And we call this the I band because it's between the A bands. One sarcomere officially goes from Z disc to Z disc. Although the Z discs themselves, so this Z disc is part of this sarcomere, but it's also part of the sarcomere to the left. This Z disc is part of this central sarcomere, but it's part of the sarcomere to the right. The I band extends over the junction between the two sarcomeres as well. So that when we look at it, we see these A bands with an H zone in the center and an M line, and then the I band here with the Z disc zigzagging down the center. Okay. And the myofibril is a whole bunch of sarcomeres joined end to end, surrounded by the sarcoplasmic reticulum with a T tubule surrounding each one, like this. If we look at it end on, we'll see that there is an arrangement of these thin and thick filaments. That the thick and thin filaments arrange around each other, that there are each thin filament, like that one right there, interacts with 3 thick filaments, and each thick filament is surrounded by 6 thin filaments. In the H zone, no thin filaments. It's only thick filaments. In the I band, in the position of the thick filaments are the connectin proteins that act as a spring. So A band from here to here with an H zone in the middle and an M line. I band from here to here with the Z disc in the middle. Like that. So we've talked about the passive tension and in connectin. And this happens right within the sarcomere. This is a protein that happens here and holds the thick filaments in place to the Z disc and acts like a spring. There's another very important protein called dystrophin, and it anchors the myofibrils to the sarcolemma. It's important in a pathology called muscular dystrophy. People with muscular dystrophy don't make strong dystrophin, and it fails quite often, making the muscles weak and progressively weak, because as they fail, we can't use those myofibrils. The myofibrils, when you think about it, are creating a force, but they have to pull on something. You can't play tug of war without a rope, and the dystrophin is what is the rope that anchors to the cell membrane which then anchors to the connective tissue, the endomysium and the perimysium and the epimysium that we talked about before. We're not going to talk about muscular dystrophy, but suffice it that it's a genetic mistake in making dystrophin. Okay, muscles obviously are going to use a huge amount of ATP. It takes a lot of energy to contract a muscle. So that means muscle fibers have to have lots of mitochondria. They produce ATP aerobically because you want to get 36 ATPs out of every glucose molecule. So that's an important thing. There has to be a real good blood supply to bring oxygen and nutrients to the muscle. But oftentimes we use muscles very, very quickly. Like you'll go kind of from 0 to 100 very quickly. So delivering the oxygen and delivering the sources of energy can be a real problem. So we should store that stuff in the muscle. So we store oxygen on a molecule called myoglobin. Virtually the same thing as hemoglobin, except that it's found in muscles. And it's red. It's pigmented, and this is why we have red muscle. Why skeletal muscle is red, because of the myoglobin. And the more myoglobin it stores, the redder and darker the muscle is, making dark meat. We also should store some carbohydrates when fuel is needed quickly. So we store glucose in the form of glycogen within the muscles. When the muscle's at rest, we make ATP and we have ATP stored. But when that reaches its maximum, it'd be nice to store a little bit more energy. So instead of adding the phosphate to an ATP, what we do is we add a phosphate to something called creatine, and we make creatine phosphate. Now, creatine phosphate isn't an energy source itself, but what it is as soon as an ADP meets a creatine phosphate, the phosphate leaves the creatine phosphate and goes to the ADP to make ATP very quickly in one step. So this is kind of like having a power bank to recharge your cell phone if you are running low on energy in the cell phone. We don't use the power bank to run the cell phone. We use the power bank to recharge the battery in the cell phone. It's the same deal. And this is why if you go to Popeyes or any of those places, that support gym membership, you can buy creatine. The idea being that if you can maximize the amount of creatine and therefore the amount of creatine phosphate in the muscles, you have more endurance. We'll see in a little bit why that doesn't work very well. It might give you a couple of extra seconds of endurance, but that's about it. But those are important. The myoglobin stores oxygen, glycogen stores carbohydrates, and creatine phosphate is a way of storing energy beyond ATP. Okay. Obviously, a single muscle will have thousands and thousands of muscle fibers, different muscle cells. And each one of them will have thousands of myofibrils, the contractile organelle. And each myofibril will have hundreds, if not thousands, of sarcomeres. And each of the sarcomeres has hundreds of thin and thick filaments. And each thick filament has hundreds of heads, myosin heads. And it's the myosin heads interacting with the actin beads on the thin filaments that cause the contraction. So all of these things, to be effective and add up to a full muscle contraction, need to be coordinated. Now, skeletal muscle is voluntary, and so that means that we can voluntarily contract it using our brains. We call the neurons that control this the somatic motor neurons, or just motor neurons, but they're really somatic motor neurons. But it would be wasteful if we had to have a motor neuron going to each individual muscle fiber, because they work together. So what we instead have is the axon of the motor neuron. When it gets to the target muscle, like the biceps or whatever, it branches. And where it branches, it will go, the axon branches will go to a bunch of individual muscle fibers. And those muscle fibers that are all bound to the same motor neuron end up then working together. Anything that signals contraction in one will signal contraction in all of the ones in this unit, in this group of muscle fibers. So we call the motor neuron and all the muscle fibers that are innervated by that a motor unit. They work functionally as a syncytium. They functionally, the motor unit, all the muscle fibers contract simultaneously. So they work as a unit. In this particular picture, these four fibers are all innervated by motor unit, looks like 2 here. And these 2 are innervated by motor neuron 1. So when motor neuron 2 fires, these 4 contract. When motor neuron 1 fires, these 2 contract. And if they both fire simultaneously, all 6 contract. Make sense? You'll also notice that in this picture, there are multiple nuclei and they're all on the periphery. And you can see the striations. You can see the A bands and the I bands through the microscope. So a motor unit, a motor neuron, and all the muscle fibers that it controls. So sometimes motor units can be very large and some of them can be very small. So depends on the amount of control we want or the amount of strength we want. Small motor units can have like fewer than 5 muscle fibers. And what it allows is very, very precise control. So, for instance, the muscles that move our eyeballs have very small motor units because we can control what we look at very, very finely. The muscles that control your right index finger, if you're right-handed, will have small motor units so that we can control very precisely what's going on. And if we need strength, we just recruit more motor units right? In muscles that we don't need precise control, we can have large motor units. We can have thousands of muscle fibers in one motor unit. Very little control, but a large amount of force can be generated quite quickly. So think of the gluteus maximus muscle. We want lots of force in the glutes for running and climbing stairs and lifting our body weight and doing things like that. But we don't need a lot of control because it's kind of an all or nothing thing. When you're climbing stairs, you're lifting your body, and then you're not. So large motor units, lots of force, but not as much precise control. And um, and to make a motor unit even, the fibers are dispersed throughout the muscle. Like, so they're not even all in the same fascicle, because you don't want the force to be concentrated in one area of the muscle. You want it to be spread out, especially for the large motor units. So they don't have to be physically next to each other, like in this picture, but can be spread across multiple fascicles across the whole muscle. Okay. Now, most muscles, the named muscles, have got a number of small motor units for some precise control that are fairly weak, and some large motor units that get recruited when we need to create a lot of force, but there's not much control. And some intermediate-sized motor units that kind of balance between these. And so a single muscle will be made up of a number of different types of motor units. So the place where the nerve interacts with the muscle tends to be someplace in the mid region of the muscle fiber. And there is a real structure. It's called the synapse, the neuromuscular junction. And there's a couple of different structures that we need to be aware of in the neuromuscular junction. There is, at the end of the axon branch, there is a widening called the synaptic knob. It's basically a widening of the end of the nerve. Within the synaptic knob are found a whole bunch of vesicles, little bubbles, as it were, filled with a neurotransmitter. In this case, the neurotransmitter is called acetylcholine. All motor neurons make acetylcholine, and all synaptic knobs of those motor neurons contain vesicles of acetylcholine. And it is in proximity to a folded area of the sarcolemma called the motor end plate. This is a specialized area, and it's all folded, so there's lots of surface area. Right? And on the motor end plate are receptors to the neurotransmitter. So there's acetylcholine receptors all along this motor end plate. In between, there is a space called the synaptic cleft, and it is continuous with the interstitial fluid between the cells. So it's filled with interstitial fluid. The synaptic knob is not physically attached to the motor end plate, but there's this space in between called the synaptic cleft. So the synaptic knob has the synaptic vesicles filled with acetylcholine, right? It has calcium pumps in the membrane of the plasma membrane of the nerve cell, and it has voltage-gated calcium channels in the membrane. So when the voltage-gated channels open, calcium goes into the cell because there's calcium outside of the nerve in the interstitial fluid and not inside. And the reason that it's not inside is because the nerve actively, using active transport, pumps calcium out of the nerve. So it establishes this gradient. Any calcium that's in the nerve gets pumped out into the interstitium. And it's let back in if these calcium channels open. The motor end plate is part of the sarcolemma, lots of folds for surface area, and has these acetylcholine receptors. There are also channels that open up that allow sodium to come in and potassium to go out. And these channels open when the receptors are bound to acetylcholine. So acetylcholine is in the synaptic cleft that binds with the acetylcholine receptors and opens the sodium and potassium channels. No acetylcholine in the cleft, sodium and potassium channels closed. Okay. The synaptic cleft is a space. Now, there happens to be an enzyme that kind of resides in that space, and it's called acetylcholine esterase. And its job is to break down acetylcholine and make it not work. So makes it so that acetylcholine is released, the channels open, then acetylcholine is removed, the channels close. The removal is acetylcholine esterase. It breaks it down. Other than that, the channels would be open all the time. It has to be removed. So it looks like this. There's the vesicle filled with acetylcholine, the little red dots. Calcium is here. Calcium's in the interstitium. It's pumped out constantly using ATP. If the channels get open, and this is a nerve firing, when the nerve fires, a signal comes down the nerve and opens these voltage-gated calcium channels. Calcium floods into the synaptic knob via facilitated diffusion. The calcium binds with the vesicle of acetylcholine that makes the acetylcholine vesicle come to the surface and release its acetylcholine into the cleft. Potassium are these dark blue ones, and the acetylcholine is the red, and kind of the clear ones are sodium. When acetylcholine binds to this channel here, it allows sodium into the cell, and it allows potassium out of the cell. Cool? So, nerve fires, the nerve signal causes calcium, allows calcium to enter the nerve, which causes the release of acetylcholine, which causes the sodium channels to open and allows sodium to go from the interstitial fluid into the muscle cell based on a concentration gradient. And the reason there's a concentration gradient is because sodium-potassium pump sodium out of any sodium that comes into the cell, into the muscle cell, gets pumped out using active transport. It comes in by facilitated diffusion. So this would be a good place to stop this video. And when I come back in the next video, we will talk about resting membrane potentials and what all of this sodium and potassium does to the cell.